Hi, everybody. My name is Jessie Ellis, and I'm going to be talking about the evolution of virulence and transmission in pathogens. For a little bit of background information, the term virulence refers to the ability for a pathogen to cause disease in a host, and the term transmission refers to a pathogen's ability to spread from one host to another host. These are the two most important traits of a pathogen, and all of the different things that contribute to the dynamics of evolution of pathogens have to do with either virulence or transmission. So some environmental factors that influence virulence and transmission of a pathogen have to do with host population crowding. Depending on how crowded in one area a population of potential hosts is, it could be more or less advantageous for a pathogen to easily transmit to other hosts. Another influence on the evolution of pathogens has to do with what patterns of contact there are between hosts that a pathogen can use to transmit from host to host. Obviously, if there are not certain patterns of transmission available in a population, then it would be disadvantageous for a pathogen to transmit using that route. Also, if there is a common antimicrobial use by potential hosts in a population, that is also something that would affect pathogens because it would be best for them to be able to resist the antimicrobial products used by the hosts of the population and still be able to infect the hosts. But if they're not able to, then they will not survive in that population of hosts. The two other things that will influence pathogens' traits is how it will acquire nutrients and how mobile the pathogen is in its environment. Now, one of the most influential factors on the pathogen's transmission is genetic variation. And what research has found is that pathogens with high genetic variation are able to evade the host's immune response, and they best survive in that way and don't need to transmit to other hosts very quickly, since they're able to evade the immune response in the host that they're currently in. On the other hand, pathogens with low genetic variation are easily identified and destroyed by the host's immune response. So for these pathogens, it would be advantageous to be able to transmit quickly to other hosts before they die out in the current host from the immune system that they're not able to escape from. Now, there are different ways that genetic variation can develop in pathogens, and one of the examples for bacterial pathogens is through transformation. The transformation refers to when a bacterial cell will take up foreign DNA and use it alongside its chromosomal DNA. Now, small circular chromosomes called plasmids are the genetic material that is transformed up into a bacteria cell. And the plasmid also has to contain some sort of gene that is useful to the bacteria cell, or it will be discarded by the cell since it's of no use. So this is an example of how bacteria can become resistant to antibiotics. They can take up a plasmid that has a gene that allows them to grow in the presence of an antibiotic. And since that gene is useful to the bacteria, that cell will keep it and use it alongside its chromosome. Now, one example of how variation develops in viruses has to do with the replication of the viral genetic material. Viral genetic material can come in either DNA or RNA form, but no matter what form it takes, the virus injects the genetic material into the cell and has the cell incorporate it into its genome to then make more virus and continue the infection. For RNA viruses specifically, they have to first create DNA using their RNA genome, which requires a process shown here called reverse transcription. And the thing about reverse transcription is that there's no proofreading activity. There's no way to correct any mistakes that are made when it's copying the RNA. So RNA viruses have a high level of consistent mutations since their genome needs to be replicated before it's used by the host, and mutations cannot be noticed or repaired. So those are some of the examples of how variation can occur in pathogens. Once a variation occurs in a pathogen, natural selection can place pressure on the pathogen to develop different traits regarding virulence and transmission. Some of the characteristics that are involved with virulence is the rate of growth of a pathogen and if it can target host immune cells. If a pathogen has a very fast rate of growth, then it often has low virulence and does not cause a very serious infection. But if it has a slower rate of growth, it's more beneficial for that pathogen to be more virulent and cause more serious illness in that host. Also, if a pathogen is able to target and kill host immune cells, then it has higher virulence because it can cause more serious harm to the host. The characteristics that are involved with transmission variation in pathogens have to do with the rate of growth as well, and if the symptoms of the disease that the pathogen causes is helpful, harmful, or indifferent to its transmission, and if it can target host immune cells. The pathogen has a very fast rate of growth. It will also have a high transmission rate because it grows so quickly. It works best at transmitting and spreading to other hosts to continue its infection. The symptoms of the disease that the pathogen causes can affect how well a pathogen can transmit to other hosts. A disease symptom can be helpful to a pathogen to transmit to another host, or it can prevent a pathogen from transmitting to another host, or it can also be indifferent and not be helpful or harmful in any way for the pathogen to spread to a new host. Now, the ability for a pathogen to target and kill host immune cells also applies to transmission, because if a pathogen can kill host immune cells, it doesn't have any need to transmit to other hosts very quickly, versus a pathogen that can't target host immune cells will have to transmit to other hosts quickly before the immune response kills them in that host. In conclusion, understanding the dynamics of pathogen evolution when it comes to virulence and transmission is very important. It's important to understand in order to develop preventative measures against new pathogens arising and causing outbreaks or even pandemics. It is also important to understand so that we can be sure not to put in place medical policies that would provide selective pressures that would create stronger pathogens, or give them the ability to adapt to antibiotics or antivirals that we would use to treat patients with infections.